Sri Sai Ram. Welcome to Sri Sathya Sai Loka Seva Gurukulam. Today we are going to start a new chapter, chapter number 21. In that we are going to study the building blocks of life. That is, we are going to talk about the cells and the tissues which is present in the living organism. We will start the presentation now. In this chapter, overall we are going to study the structure and function of cell, how the cell divides and how the cells combine to form tissue. We will study about stem cell technology, that is latest and emerging technology, thereby we can replace the damaged tissue, that is we can repair the tissue. All those things we are going to study in chapter number 21, and today's learning objectives are how the cell was discovered in a living organism and what is cell theory. We will study the structure and function of the cell. Later we will see the type of the cell as prokaryotic cell and eukaryotic cell. So, we will start with our lesson today. You know about this figure, right? This is the structure of a microscope. Have you ever seen people constructing a house? The mason and the construction worker, what they will do? They will keep one by one all the bricks, right? And finally, we will get a beautiful house. Similarly, in our body, we have multiple of cells. All these cells originated from a single fertilized egg. Only one cell which divides and it is forming many cells. Many cells, then again it divides and it is having some differentiation and it is becoming tissue. Then few tissues combine and it forms organ, and organs combine to form organ system, and organ system combine to form a living organism. We do have many different kind of organ system which is present. So, the basic thing what we have to study here is how these organ systems are made up of. Here we are going to start with the cell, the very small part of the organism. We will start seeing about this chapter now. The cell was first discovered with the help of a microscope. The microscopes are having many lenses present, and these are known as objective lens. Since it is going to nearer to the object, here only we will keep the object, and then we have the eyepiece on the top. Through that we can able to view it, and this is a common kind of a compound microscope. You may have handled small magnifying glasses and all when you are a kid, right? So, it is very much similar to that, but instead of having only one magnifying glass, you can imagine multiple magnifying glass. With that you try to make it to focus. Okay, similar to that, here it is. So, this is the major breakthrough for discovering a cell. Robert Hooke discovered the cell in 1665. He was the one who discovered the cell because he has an opportunity to have his own microscope to be designed, because his father was working in one such place where he can grind many lenses. Thereby, Robert Hooke explored the world. Whatever he come across, he observed it under microscope. First what he did was, he took a piece of cork and then he made a thin section in the cork and he observed it under the microscope what he has created. And he could able to see so many tiny packets and compartments which was present inside the cork tissue. So, he could able to tell that it is a cell. Cell you may know, right? Cell is small rooms which is present in some of the jail and all. Cellular jail you may be heard of in Andaman Nicobar Island, right? So, the cells are small compartments which is present in consecutively, that is so many kind of tiny compartment which is combined together, it is present in one place, right? So, similarly here they could able to see small compartments which is arranged next to each other, and hence he coined the term cell. These were like honeycomb structures, that is next to each other. You must have seen a hexagon honeycombs, right? So it is all similar to that. Now we will see what cell theory is telling about. The cell theory was formulated by a German biologist. There were two German biologists who coined cell theory. The first one is M.J. Sheldon in 1838. The next person is T. Shivan in 1839. Separately, they formulated cell theory. Here you can see the cell and its nucleus. Okay, this is the structure of white blood cell and it is stained. Okay, to give color, they will make some arrangements so you can able to see, distinctly see the cells and in internal constituents and the outermost boundary. Okay, this is a white blood cell. What cell theory is telling? Cell theory states three important points. The first point is cell is a structural and functional unit of living organism and it is present in all the organism and all the organism is composed of cell. Second, all new cells arises from division of pre-existing cell. We cannot able to create a cell with inanimate things. The third is the function of the organisms are the outcome, that is the effect or the benefit of combination activity and interaction between the cells. All the cells has to interact with each other, communicate. What each cell has to do is communicated by each and every cell's neighbor. Okay, so that's what it is coined. So these are the three important points of cell theory: structural and functional unit, and cells arises from pre-existing cell and they are working or they are functioning by the combination activity and the interaction between the other cells. So what is a cell? If a question is asked to define a cell, what we can write? The cell may be defined as the structural and functional unit of living organism which is capable of independent existence. Here you can see a red blood cell moving in the blood vessel. Inside the blood vessel, how red blood cells are moving. You can able to see the red blood cells, so many are floating, right? There are three basic parts of a cell. What are they? The first one is the cell membrane. Cell should have a boundary and in that boundary certain chemical compounds are present. By that we are getting that boundary, that is known as cell membrane, that limits the cell and gives its shape. Limits means where it has to stop. Okay, the cell, when we are drawing, we have a compound wall, right, around our house, like that. The cell should have something to demarcate, like where I am, okay, how I am, all those things. And then it gives a shape to a particular cell. We have seen in the before slide red blood cell, right? It is all circular. You can able to see like little suppressed sphere shaped cells. Second is it has a genetic material, that is DNA, which is present inside the nucleus. The third part is the fluid part of the cell, that is known as cytoplasm. Cytoplasm has very many different kind of cell constituent which is present, that we will see in the forthcoming classes. So overall, when you are seeing the cell should have a boundary, it has the cytoplasm and it should have a nucleus which is present. Okay, all this mitochondrion and other structures we will see elaborately later. There are two different kind of cells which is present. The first kind of cells are known as prokaryotic cell and second kind of cells are known as eukaryotic cell. I repeat, prokaryote and a eukaryote cell. Okay. So here what a prokaryote cell will look like. Prokaryote, we may be knowing about bacteria which is causing some kind of infection. Here you can able to see this is a prokaryotic cell where the nucleus is not prominently present here. The DNA, that is the genetic material which is just present in the cytoplasm. It do have cytoplasm, it do have other cell organelles, but we can able to see that it is not having nucleus. The genetic material DNA which is present just loosely in the cytoplasm. In eukaryote, we can able to see a proper nucleus, and inside the nucleus we can able to see chromosomes. In the chromosome we can able to see the DNA structure. So that is the basic difference between a prokaryote and a eukaryote. Pro means previous, okay, initial, and karyon pertains to the genetic material. Here U means true, eukaryote, true kind of DNA which is present, okay. That is the difference between a prokaryote and a eukaryote cells. So we will see now how a prokaryotic cell is organized. Pro means before and karyon pertains to the nucleus, and they do not have well organized nucleus. That is the first point for the description about the prokaryotic cell. The genetic material is single stranded DNA which just lies in the cytoplasm. You can able to see that blue color strand which is present in the cytoplasm. Here it is known as nucleoid. Okay, nucleoid. What is nucleoid? The DNA, that is the genetic material which is just dispersed in the cytoplasm. And nuclear membrane is not present. There is no boundary which is present between the DNA, the genetic material, and the cytoplasm. So there is no nuclear membrane present, and cell organelles like mitochondria, lysosome, endoplasmic reticulum, chloroplast, nucleus are also absent. It is not present in the prokaryotic cell. Okay, you can able to see the cell is very simple, cell membrane is there, otherwise it is having various other kind of things which is present. It need something to do protein synthesis, so ribosomes are present, and it has some food material which is stored inside the nucleus, that inclusion bodies we can able to see. Apart from the genetic material, we can able to see plasmid which is present. Plasmid is a extra chromosomal material that is also present, and outside the cell you can able to see tiny hair like structures, right. These are known as fimbriae, and then a very long tuft which is present as flagella. These are the various things we can present outside the cell. Okay, this is how a prokaryotic cell is organized. Okay, here we are considering this bacteria and as an example. Bacteria, blue-green algae are belonging to this category. In eukaryotic cell, what are all the various things we can able to see? In eukaryotic cell, we can able to see the nucleus very prominently. Eukaryote means true, and then true nucleus, when it is present, it is known as eukaryote. The DNA is present enclosed in a nuclear membrane, and it is forming a nucleus. The genetic material is made up of DNA only. The DNA is present as a network, and it is known as chromatin fiber, and which is getting divided during the cell replication. The cell is producing two cells from one cell. The membrane bound organelles such as mitochondria, endoplasmic reticulum, lysosome, chloroplast, nucleus are present. Okay, that is the major difference between a prokaryote and eukaryotic cell. And here examples are plant cell, fungal cell, protozoan cells like amoeba and all, and then we can able to see in animal cells. Even though in our human also it is belonging to this eukaryotic category. That we are coming to an end, and I have some home assignment questions for you. Who discovered the cell? Name the instrument which helps us to see the cell. Who formulated cell theory? Explain cell theory. Differentiate between prokaryotic and eukaryotic cell. We will come to a conclusion of this chapter. We will see the quote of Bhagwan Sathya Sai Baba. Let each and every cell of your body be filled with His divine name. That is the quote given by Sathya Sai Baba. What do you understand from that question? Each and every cell has to be filled with His divine name. With this note, we'll conclude today's session. We'll meet you again in the next session with this answer. Thank you. Om Sri Sai Ram.